congratulations for making it back so early. Yeah. Today's session is on emerging techniques to study organelles, and it's a great pleasure to have Professor Sandy Patel, Professor of Cell Signaling at UCL, to chair the session. Thank you.
Great. So, Anthony, good job getting everyone together for a really exciting meeting. First day, I think, was really informative. I hope the rest of today will also be equally informative. So, yes, I'm based at London. My interests are in lysosomal calcium signaling. Yesterday, Maggie gave us some of the work that we do. I actually got interested in that area by working with Anthony Daglieri here many years ago, or as I like to say, when Anthony worked for me. So let's get started. So Fran was telling us that one way of pretty much innovating is to bring academia and industry together. So this first session we're going to have an academic talk, followed by an industry talk. And our first speaker is Teresa Alonso from the University of Valladolid. So Teresa is well known in the calcium signaling field. She makes measurements of calcium not in the cytosol, but actually within organelles, which we're all interested in. So she's hopefully going to tell us about her historical work and then maybe some new stuff.
Yeah.
Teresa, 25 minutes.
Yeah, thank you, Sandeep. I would like to start by thanking Anthony and Francis for this very special meeting, actually. As you said, I think it's the first time, and when I received the invitation, the title was already a bit surprising, no? I mean, pharmacology and organelle was the first time. So I tried to, and Anthony asked me to talk about the calcium sensors, luminal calcium sensors, he said. So that's what I did. So that's why the title is a bit ambiguous, and because luminal has more sense in high calcium concentration organelle. So that's what I'm going to do. So by now, after the whole session, the whole day yesterday, we all are convinced that the cytosolic calcium peaks. Can you see? Yeah. Rises from 100 nanomolar up to 1 to 2 in a normal cytosolic environment, not in a microdomain. This peak is shaped by organelle. So then organelle act as sources like the ER by releasing calcium through IP3 receptor or ionodyl receptors, also the lysosome. And by acting by source and by sinks, no? By uptaking calcium through CERCA or other mechanisms like in the mitochondria by the uniporter. So at the same time, organelle regulate enzymes, resident enzymes that are regulated by calcium in the organelle. So disturbations of this organelle calcium dynamics are implicated in many, as we have already heard here, pathological conditions. So that's why the title of this slide is why to bother about calcium within the organelle. It's not enough to monitor calcium, cytosolic calcium. Well, I mean, it's obvious that no, it's not enough. So the first slide is our main tool that we have been using for many years. And this aquarium. Aquarium is the champion of all the. It's not so well extended as now the GCAMs or whatever, or the Fura2, of course. But it has the triple merit to be the first calcium indicator. Many people don't know this, but aquarium was purified very painfully. I invite you to read the story of Shimomura, Nobel Prize in Chemistry 2008, because his life, personally and also professionally, is fascinating. So he very painfully separated and purified aquarin from jellyfish and separated from green stuff, as he called it. The green stuff, it was the GFP, and it was, ironically, he got the Nobel Prize for the discovery in the green fluorescent protein, not in the aquarin. So aquarin is a very small, 25 kilodaltons oxidase, and this protein has the ability that binds 3 molecules of calcium when it is reconstituted with selenteracin and oxygen and emits blue light, and the selenteracin gets oxidized to selenteramine. So, as I said, the first thing that Shimomura purified this protein and very generously sent it to many laboratories, and they started with injecting in the cells, and in the barnacle was the first thing. Then they cloned the gene, and the gene, the recombinant protein, was the first GETHI that was used. And finally, through the work of Tullio Posan and Rosario Rizzuto, they targeted, first GETHI targeted to an organelle, which was the mitochondria. So along these years, we have monitored calcium in different organelles, and here are the traces, and we have published papers that, for example, the nucleus at that time, it was important, or the Golgi dynamics, also controverted, and more recently in the endolysosome. So this was one of our first work in mitochondria, where bovine chromaffin cells were expressing mitochondria targeting aquarin, and we could record at this time very unexpected, very high levels, 300, 400 micromolar of calcium in these cells, and this peak was in intact cells. Upon depolarization stimuli, high potassium, and this was completely blocked with the protonophore CCCP, and it was reversible. So the cells were perfectly well. And we found that this high amount of calcium was only being uptaken in mitochondria that were close to the plasma membrane in a sort of microdomain of high calcium, close to the secretory vesicles. In fact, this mitochondrial uptake had an impact in catecholamine secretion, because these are catecholamine secretion peaks with high potassium, and when mitochondrial uptake is blocked, then the secretion is much higher. So, whereas there are other mitochondria that they are deeper in the cell that didn't see this high amount of calcium but much lower levels. Because this is Organelle Pharmacology meeting, I brought this slide that I normally don't put in the talks, but this was a collaboration that we did with Fabiana Perocchi. So we put together our efforts. She did a screening in GIST, and we did it with a tool that is HeLa transfected with mitochondria aquarin in permeabilized cells. This is important for as a drug screen high throughput assay, looking for drugs, specific drugs for inhibitors for the uniporter, MCU, as Christina mentioned yesterday. This was also a screen. And so we screened like 700 drugs by this assay, and we found mitosantron as a very specific inhibitor of the uniporter. So this is an example how these two basic science in organelle and calcium sensors can be used for screening important functions in cells. In jellyfish, what it really happens is that aquarin, when it is reconstituted with its cofactor and is activated, then it emits green light, not blue light as expected. There is a shift in the luminescence spectra from blue to green. And this is due to a phenomenon that occurs in vivo in the organ of light, in the umbrella of the jellyfish, where these two proteins, aquarin and GFP, are very close together. And they suffer bioluminescence resonance energy transfer between these two proteins. That's why Shimomura had so much problem to separate the two. Because functionally they work in the jellyfish to emit a light. So what we have done in the lab is to mimic nature and to fuse together these two proteins. An old version of GFP and apoaquarin with a linker of 16 residues. We call this protein GAP from a GFP aquarin protein. And this calcium indicators family has the ability, is unique in its sense because it has the ability to operate in a dual mode as a fluorescent indicator without cofactor. Then one can measure GFP fluorescent in organelle. This is an example of calcium imaging in astrocytes in the ER, and these are several pulses when GAP is targeted to the ER. So this is in this way, aquarin only provides the calcium binding elements in the same way as calmodulin or troponin or any calcium binding protein. In this way, there is a conformational change by calcium binding that is transferred. We still don't know exactly how, but to the GFP moiety, and then the GFP is sensitive to calcium. So the second mode is luminescence, which is the standard way for apoaquarin. But in this case, you have the GFP there. In this case, GAP works, it has to be reconstituted with selenteracin. In this case, it's very hard to do imaging, although it's possible. And so we routinely monitor calcium in cell batches. So this mode has advantages in certain conditions, in certain organelles, in contrast to fluorescent mode, especially in small vesicles like Golgi or endolysosome. And here are the advantages of the luminescence mode. It doesn't require citation, so there is no phototoxicity. The dynamic range is much higher than the fluorescent, normally low background, so there is a very good signal-to-noise ratio, very low calcium buffering, and it's less sensitive to pH normally than the fluorescent proteins, and we can do recordings up to 1 hour. So in the rest of the talk, I'm going to give a couple of examples of GAP targeted to the ER or sarcoplasmic reticulum. And in the second part, I will focus on the luminescence mode by targeting GAP to the endolysosome. So first, fluorescent GAP. This is a 50 kilodaltons protein, of GFP and aquarin, and so it has two maximum of excitation, one at 400 and one at 470 nanometer. When calcium binds to GAP, the shorter wavelength decrease its fluorescence, and the longest wavelength increase its fluorescent intensity. That means that this is a radiometric GETI, so no cofactor, and this is of course very advantage for measurements, as we all the people who do calcium measurements know. Radiometric is advantage, and especially for doing something that I'm going to show in the next slide, resting levels of calcium. So we have mutated GAP, and so we have different versions with different affinity for calcium. So we have one which is high affinity, 0.2 micromolar, very optimus for cytosolic or nucleus, and then lower affinity, one moderate, 12 micromolar, and the ones that we use for the sarcoplasmic or endoplasmic reticulum, which is 500 micromolar, GAP 2 or 3. The gold standard for a new calcium sensor is performance in vivo. Because this GAP is a fusion of two proteins from jellyfish, it doesn't have endogenous partner. It doesn't have endogenous homologs in mammalian tissue. So we could make a transgenic mice where GAP targeted to the ER is under ubiquitous expression. Now we have a conditional one. And that the advantage of having this mice is that one can measure directly calcium in ex vivo in intact organs. So we did that in hippocampus, in pituitary gland, and in pancreatic islets. These mice are available in the ENMA. And also another criteria that is very important is that the GETI must work in vivo. So for in vivo studies, we choose the Drosophila system. So we targeted GAP3, targeted to the sarcoplasmic reticulum in muscle cells, so under the promoter myosin heavy chain. So here is a fly that only the muscles in the thorax express the protein. So we, because it's a radiometric sensor, we can monitor calcium in the thorax directly, lives whole intact, no, anesthetized, but but the whole. So we can monitor 470 and 405 nanometer, so by doing the ratio. And then important, we design also a method to calibrate, which is very, very hard, but it's important to calibrate in calcium. So we, in each measurements, we record the R min, the minimum amount, the minimum fluorescent with low calcium. Then we ask the question. So we all know that calcium is very important for contraction in the muscle, and we ask along the lifespan of the fly, from 7 days up to 35, which are very old flies already, is this calcium in the sarcoplasmic reticulum altered, or is it varies? Because there were some reports already that in the cytosolic calcium there were alterations and it was altered. But we thought maybe it's not the cytosolic calcium, and this one, once more, an example of going deeper and going to the organelle who is the origin of the hypothesis. So we tested if the sarcoplasmic reticulum resting calcium was altered along the lifespan. So as you can see, there is a drop, a progressive reduction in the ratio and in the calibrated calcium along the lifespan. So young flies have much higher calcium than old flies. So because we have this calibration, we can measure directly the calcium concentration. And this calcium we correlated with the muscle function. So we did that with a standard method in Drosophila field, which is the climbing. I didn't show the video, but it's very dramatic. So you put the cells, the flies on the bottom of a tube, and then you leave them a time, and you wait till they can climb along the tube. So by doing this, we could see a very nice correlation between the levels of calcium in the sarcoplasmic reticulum and the climbing ability, which is a proxy for muscle function. So there is like a threshold of around 300 micromolar in the sarcoplasmic reticulum, which are the young flies, but then older flies start to reduce their calcium, and this compromises dramatically the climbing ability, so the muscle function. So they have sarcopenia. So the rest of my talk, I'm going to concentrate on endolysosome. So endolysosome is a real challenge organelle. We all know that because of the pH, of the calcium, of the degradation enzymes. So a luminal indicator should be well targeted to this organelle, so specifically target should be pH resistant, so should have low calcium affinity because the expected calcium concentration is very high, it's like the ER, and ideally it should be able to be calibrated. So we took our gaps, so GFP-acquiring proteins, two versions of this, and we fused them with BAM7. BAM7 is a protein of the SNAR complex that is located in the late endosome and secretory lysosome. So BAM7 has a domain that is cytosolic, one transmembrane domain and one very tiny portion in the luminal part of the endolysosome. And there we hook the gaps. So to these sensors, we call it ELGA. Actually, my friend Sandeep named it. He's very good for giving names. So ELGA is endolysosome gap, but gap ELGA sounded much better than ELGAP. So there are two versions. So these are low affinity for calcium, both of them, but one is lower affinity, so we can measure in the range of hundreds of micromolar up to millimolar. But this is low affinity, and it allows measurements during 1 hour. These ELGAs have a pattern when transfected, punctated, as one expected for the endolysosome system. They co-localize with endolysosome markers like LAMP2 or TRPML. And here are two amplified lysosomes decorated with ELGA that express also TRPML in super resolution. No. So because of the expected location of BAM7, because the ELGAs are visible in live cells, which if it would be at 4.5, they would be quenched. So we think we are not targeting the lysosome very acidic, but mildly acidic, so late endosome. so at pH 6. So we did a calibration of LGAT1 with at this pH 6, and it's very similar. 2 minutes? Oh, wow. Okay, so the uptake of calcium in this compartment we could measure, and is around 377 micromolar in this compartment. It's ATP dependent, this calcium uptake. It is tapsigargin sensitive, so it means that it accumulates calcium through an active mechanism. So because it's co-localized with TRPML, TRPML can be activated, as we heard yesterday, with MLSA1. MLSA1 produces very robust and reproducible release of calcium with an EC50 of 7 micromolar, and this is pharmacologically inhibited with the inhibitor MLSI1, and also the other endogenous agonist, PIP2, releases calcium very nicely. And this is both of them are blocked in the genetically by transfecting the TRPML death pore mutant. Other TRPML family isoforms, like 3, are also activated through this mechanism. So the most important slide of this talk is this one. So NADP can release calcium reversibly and robustly when it works. So around half of the trials don't really work. But when it works, it's very nice, and you can see in HEC or HeLa cells release in. These are not overexpressing TPCs or anything, just normal HEC or normal HeLa, and we see a release of calcium through NADP. And finally, unsurprisingly, we saw in this compartment a release of calcium through IP3, which is blocked with heparin, and in the triple knockout of IP3 receptor. HEC cells, they don't see any release at all, and it can be rescued by IP3 receptor 1. So we did all the analysis of immunofluorescence and also of EM to localize IP3 in endolysosome, IP3 receptors in endolysosome. So our current model is that we have targeted an organelle, mildly acidic of pH 6, which has around 400 micromolar calcium, and it uptakes calcium through an active mechanism, and it can mobilize it through TRPML, TPCs, or IP3 receptor. And this calcium can be used for local activities, local functions, but then it can, in trans can be amplified through the IP3 receptor or IML receptor from the ER, acting for more global functions. So, and finally, people who has done most of the endolysosome work is Belén Calvo and Patricia Torres, and other members of the lab previous, and our great collaborators, Sandeep, David Jewell, Kristen Green, and Vera Moisenkova, and the reagent shared by Colin Taylor. Well, I didn't show that, but it's a cell line, HeLa, that is expressed IP3 receptor endogenously with tagged with EGF. Thank you very much.
Okay, we've got time for a couple of questions. Yes.
Regarding your data of the sarcoplasmic reticulum calcium in the fly,
yeah.
Why is it that, do you know why the levels decrease with age? Is there less uptake, more release?
Yeah, so we follow a bit this, and we try to we check if there were less IP3 receptor or less CELCA, or so we couldn't really. I mean, there was in this some of these players are less expressed in the old flies. but that couldn't explain these high differences, because it's like from 300 up to 50 micromolar. So it couldn't explain the difference that we saw. So we saw a twofold decrease in levels of expression, but that couldn't explain the huge amount.
Yeah, I'm very intrigued about the lysosomal calcium sensor in the brain. And I was wondering, do you use the one type of GFP in this kind of chemical construct?
No. It's a very old version. It's called C3. It's one of the oldest versions, and it has 2 maximum of excitation.
What is the PK of this sensor?
It's around 6.
So it's not surprising that you can see only in a sort of not very acidic compartment,
Right.
Okay.
Yes. I was going to ask a silly question. Have you tried sending your flies to the gym?
Yeah, we try. Not to gym, but we make them exercise. In fact, yes. We did that. We didn't see a lot of difference. It was not the easiest experiment, but yes. And we try another experiment, which was this albastine, this drug is called S106 from Mark's lab. He has trials on this. This ryanodine receptor is a post-transcriptional modification, so you have the ryanodine receptor leaky. So we and this drug inhibits. So that's why they are using this in trials for humans. So we did this many experiments. We didn't get it to work.
One quick last question.
I was wondering whether you tried measuring in the neuronal ER, as it has like an axonal ER can be quite long, and then it's mostly smooth ER in the axonal part, which usually just focuses on calcium and the lipids. I was wondering whether you could see different flux from neuron and the sarcoplasmic reticulum.
Yeah, I mean, we tried this. Of course, it's not so easy. We have monitor slices in the hippocampus, but we didn't really pyramidal cells, but we didn't really check along the axon and the differences. We tried to do it with two photon. We have some measurements, but they were not so fine to define differences along the axon, which would be great. Yeah.
Okay, let's leave it there. Thank you very much, Keith. Okay, great. So our next speaker is Marcus Rapedius. I think I pronounced that correctly. So Marcus is a good example of a sort of in-between academia and industry, because he actually ran an academic lab before moving to nanotechnologies. He's an ion channel expert, and he's going to tell us about some of his measurements in industry.
Thank you very much, Sandip, for the kind introduction, and thank you very much also for being here, inviting me, and that I can be here, talk to you guys, and get to know you. I'm really enjoying the meeting so far. Okay. So that's me. Yes, I'm a patch clamper, electrophysiologist with the company Organelle Technology. You might have heard about, and I'm actually have been working a lot with plasma membrane, but I've been also driving actually the internal development at Nanion to provide organelle assays, because we are convinced that organelles will play a very important role for future medicine, and we want our devices to be ready, and I'm very happy to share some of the developmental work that we have done with us. So just a brief background on the company that I'm with. So the company is actually quite old already. So this very nice gentleman here, Niels Fertig, founded the company more than 20 years ago, with launching the Porter Patch, our first product. And he was a startup from the University of Munich. So we are still quite academic oriented, but we at the moment have 140 employees across the world, with subsidiaries in Japan, US, and China. And the headquarters is in Munich, Germany, and that's also where I'm from. So what we offer is expertise, and expertise combined with actually 14 devices that we can use for targeting research on membrane physiology from different angles. So we have our cell analytics branch that, with the cardiac site, flex site, and atlas, that is used for contractility, field potential recordings, and impedance, and also transepithelial electrical potential with the atlas. Then we have our membrane biophysics branch with the orbits, where you can measure high throughput single channels, and then there's also in artificial bilayers. So then we have the surfer families that can be used for direct measurements of transporters and pumps, but also ion channels, and I will come back to that. Then we have the Vesicle Prep Pro. You can use it for automated generation of liposomes. And then we have our biggest branch is the automated patch clamp devices, where you can measure patch clamp from whole cells, and we have them at different throughputs and level of automation. And the flagship is the SyncoPatch, and that is the device that I have used for the development of the organelle assays. So what kind of methods are available to functionally characterize lysosomal ion channels? And I have made here this little comparison slide, and I think you recognize what I've shown here is basically the manual endolysosomal patch clamp, where you actually use an isolation pipette to pull out an enlarged lysosome from a single cell, and then you actually use a second pipette to patch this lysosome. This pretty clever approach was published in 2017 by Chen et al. And it's actually the gold standard for when it comes to compound tests. But the problem is, or it's very challenging, technically challenging, and the throughput is also very low. So what is also used very frequently is targeting the lysosomal channels to the plasma membrane. That allows high throughput drug discovery. You can either do it with fluorescence, basically with membrane-sensitive dyes or calcium transient in a method called FLIPR, or you can measure the channels in the plasma membrane with high-throughput patch clamp devices such as the SyncoPatch. And both methods actually allow a very high throughput with up to 3,500 recordings a day. But it's a problem because the lysosomal channels are truncated and they are in an artificial environment, because the membrane lipids and the proteins in the plasma membrane vary a lot from the ones that you have in the lysosome. The third method is actually solid-supported membrane electrophysiology that we have implemented in our surfer devices. And it's very established in many publications, and it goes back to actually a gold sensor onto which you can attach lysosomes to via a tile layer and a lipid layer, and you measure an array of lysosomes in which you use a fast application system to measure your target of interest, in this case TRPML1, and then you provide a concentration gradient of your ion that you want to study, in this case calcium, and will be translocated into the lysosome, and via capacitive coupling you can induce a current in the measuring electrode, and that is then detected by the amplifier as a transient current that you can see down here. Sorry, it's a bit small, but it's calcium dependent, and you can dose dependently inhibit it with MLSI3. For the preparation, you can actually use, you do membrane preparation from cell culture cells or in, any other primary cell. You do ultracentrifugation, and then you can actually add the samples to the sensors, or you can freeze it down for later. And as this device was actually developed for transporters and pumps, you can also have a look at lysosomal transporters, for instance, the amino acid transporter cystinosine, or you can do mitochondrial preparation and look at respiratory chain complexes via application of NADPH. So the last method that I want to talk about, that's actually the novelty, will be the focus of my talk, because together with our partners, I think we have developed a method that can actually overcome the problem of plasma membrane targeting and allow drug discovery in native lysosomes. And I'm going to show you the validation that led us to believe, yes, we can really do that with something like 1,500 to 2,000 recordings a day. This would have not been possible if we would not have interacted so closely with Orion Bioscience, because they provided the highly pure lysosomes to us that we have used for the study. But we also teamed up together with Christian to provide an additional method of isolation for lysosomes via differential centrifugation, and that will be the second part of my talk. But first, I want to briefly introduce you a bit to the device and understand how it works. So this is basically the synchro patch, and behind this blue table you have the 384 channel patch clamp amplifier, and on top of that we have actually a liquid handling robot that can serve solutions, compounds, antibodies to the lysosomes into this recording module where actually the recordings take place. And down here you can see the deck space, how it is orientated in the recording. You have here the module. You have here a cell hotel where you can place your sample. Actually, it's a lysosomal hotel in this case. And you have this little white solution reservoirs, and the liquid handling head will take out the solution and move it into the module. Very important is this gripper, because the gripper can move around compound plates and consumables, because you can also use the system unattended. It means it can do recordings for you overnight, for instance, if you provide samples for it. Crucial part, of course, is this consumable that will go into the measuring module. It's a 384 chip with 384 little recording units, and each of these glass-bottomed plates has a little tiny hole onto which the lysosomes actually can land, attach to, and then you can actually exchange the luminal solution and also the extracellular solution and apply compounds. We attract the lysosomes with suction from underneath that will attach to the hole, and then you form a stable complex. And when you do additional suction, you can actually go into the whole lysosome configuration that is associated with an increase in capacitance, and you can start your recording protocol applying just a ramp here, and you see a TMM175 activated with an agonist and then blocked with 4AP. These specialized consumables that we have made for the synchro patch are also available for our other devices. Would be the Porto patch, the Patchliner M. That is, we have recently launched this guy. It's a very small and very fast unit for up to 8 recordings, and the Patchliner is a tabletop recording with full automation and up to 48 recordings. Okay. Going back to the synchro patch, I have here a little video of a real recording, and this is the samples from Orion, how they come to us. It's a small vial that contains millions of isolated lysosomes, and we basically take the sample, we dilute it and put it into one of these reservoirs. We put it onto the machine, the lysosomal hotel that is temperature controlled, and after the priming of the chip, the liquid handling robot will put them onto the chip so they can land on the hole. This is the software, and the change in color is actually showing you that a lysosome has landed on the patch hole, which is associated with an increase in resistance. Then you do the GigaZ formation, you can apply a ramp protocol, and that's actually the 384 software display from A1 to P24. And the green color actually matches the sealing quality. And in this recording, we have actually used TRIPML1 and applied MLSA1, and you can see the TRIPML1 current popping up in many of the wells. Okay, I know it was quite fast. That's why I have here like a step-to-step guide, and I would like to guide you through an experiment. So what the experiment always starts with is adding actually the lysosomes onto the holes. It's what we call lysosome catch. And you see here it's 6 different recordings, and we are catching around 90%. The next step is actually to perform the gigaseal and do like the whole lysosome configuration. And here we are down to 80%, which is like 300 lysosomes catched and sealed. But then there comes actually a very important part, because when you have to manage big data, you have to define quality control filters in order to decide, is my recording good enough for analysis, or do I want to discard it? This is the only way that you really get an unbiased view on the data. And once you have defined that, you can apply it to all your data sets. And these are the parameters that we have defined for this assay. Basically, just having a stable seal, having good enough access, having large enough current, and very importantly, have the control of the current at the end of the recording to actually say, okay, it's not a false positive that we are seeing here. When I do this on the data, I'm down to 184 lysosomes patched at the same time under exact the same conditions, qualified for the analysis. And we can do dose response curves, applying different concentrations of MLSA1 and also do the full block. And in total, this takes about 15 minutes. And the analysis can actually be automated. Okay, and here is the IT plot actually from an average trace, so it's a summary of many wells together. And you can clearly see the concentration response, the concentration dependence, 0.3, 3, and 10 micromolar, and then we have another saturating concentration to do in-well normalization. And we have the block by MLSA3 at the end. And on the second axis, you can actually follow here the seal resistance that is very stable for the whole recording. When we normalize the data to capacitance, we can see a significant increase upon the application, upon the concentrations applied, and we can fit the data, and we get something like an EC50 of 5 micromolar for MLSA. When I repeat this on the same day and also on different days, I get very similar results for the EC50 here in the range of 3 to 6 micromolar. You can see the blue dots, which is actually pretty much in line with what has been put forward by Metron Bioscience on their website from manual endolysosomal patch chemical on the same assay. And you can also in green see here the success rate is very stable, around 45 to 55% roundabout. And here is actually a comparison to other assays that can also be run on the system. The first one is lysosomal TMEM 175 assay, red blood cells, primary cardiomyocytes from pig, iPSC-derived neurons, and the best success rates you get with normal cell culture cells like NAF1.5 or ligand-gated alpha-7 recordings, and we can also use the system for mechanical stimulation like piezo channels. Now, as we understood that the system can really do this, and it's really reproducible and robust, so we tried to explore a bit the features of the system if we could apply them to lysosomes. And a very nice feature of the system is that it has a very efficient temperature control. And the temperature control operates from 10 to 40 degrees, and it also helps you to actually record always at the same temperature, regardless of summer, winter, spring, or autumn. So this is very important, and temperature can be very relevant for a lot of ion channels. You can see here the Herb channel is just giving bigger currents and faster inactivation, and also compound affinity can actually be affected by temperature. On the other hand, also cold can be a crucial factor for channel activity. So the TRPM8 is actually the cold sensor in man. And when you actually cool the module from 21 to 12, you see an increase in current. And you can also follow that over time here. You go from 21 to 12 during the recording. you increase the current and then you heat up and the current will go down. So we wanted to know, okay, can we use that for lysosomes? Are the recordings stable enough? And actually, yes, they are. You will see later, sorry. We started at 21 degrees. And for this study, we used TMM175 overexpressing lysosomes from Oriya, and we can block the currents at both voltages with 4 AP. What I wanted to point out a bit is this mixture of DCPIB and TUG, which we found out actually very nicely that it is more potent than the individual compounds. So it's really it was a really nice tool compound for us. So we compared it on one plate, different compounds, and we found this combination actually to be as potent as twice the concentration of the other compounds. So it was actually postulated in this paper that there might be some synergistic effect, but for us it was more like, oh, it's cheaper and it does the same stuff, so we keep it. Because sometimes you also have solubility issues with high concentration of compounds, so it's really nice. So we keep using it, and here's the trace at 35 degrees. It looks very similar to 21, and when I plotted the current densities, what we actually see is we see higher basal, significantly higher basal current, as well as significantly higher agonist-induced currents, suggesting that maybe TMM175 is also influenced by temperature. What we have ruled out is a change in capacitance. So the capacitance is similar at 21 and 35 degrees. So the temperature does not affect the size of the lysosomes, which is interesting. We then did concentration response curves at both temperatures, and we could actually see a very similar EC50 for the mixture and no shift in IC50 at higher temperature, indicating that temperature could indeed be an additional factor for the activity of TMM175. When we do the same thing with TRPML1 and MLSA1 activation, it seems to make not a difference, which is actually quite interesting because some of the triptans are very temperature sensitive, and we have already planned to also do cooling. Maybe cooling is activating it. I don't know. Anyway, that was actually the data I wanted to show you from Oriya. I would now switch to the second part of the talk, where we teamed up with Christian to actually provide a second isolation procedure via differential centrifugation, and that should actually be the groundwork to move on and go into more primary systems, like isolate lysosomes from immune or cancer cells, and maybe also not depend on overexpression and look at endogenous expression. Exactly. So what we have tried goes back to a paper from Sheila et al., and we have modified the approach. Basically, it's just smashing up the cells and getting rid of the debris and unbroken cells. Then we do 2 centrifugation steps, and then actually we precipitate the lysosomes with a high amount of calcium. And then what we did in the end was filtering off the lysosomes to get rid of large aggregates and particles. And we have used Christian's stable TPC2 cell line for this, so we could also actually look at the preparation step by step via fluorescence, and you can see here probably expected the unfiltered sample has a lot of large aggregates, but the filtering turned out to be really helpful, so you get a much more homogeneous, cleaner sample when you filter it, and vesicular structures here visible. And to further confirm that, actually, we did confocal imaging, and we could co-localize the TPC2 fluorescence with lysotracker staining, and you see we do have actually lysosomes in the preparation that also have integrity and activity. In the same time, and then importantly, we also tried to determine the size distribution of the lysosomes, and we found that most of the lysosomes are actually bigger than 1.5 micrometers when we use Vaclin for enlargement. And very interestingly, when we do the same quantification of lysosomal size from the Oriya preps, we actually get the same distribution of lysosomes when Oriya uses vacuuming. So the size should be big enough for patch clamp. In parallel, we also try to estimate the purity of the samples doing some Western blots. And you see here some examples. And we also introduced Appli mode because it's also a second enlarger in the endolysosomal patch clamp. And you have here the quantification of the Western blots. And we checked all the impurities from plasma membrane, nucleus, ER, Golgi, mitochondria, and so forth. And what we can see is that we do enrich lysosomal markers like LAMP1, RAB7, cathepsin D, that you can also realize here with the orange bar because it's normalized to LAMP1 in the final sample. And it works really well for plasma membrane and nucleus and Golgi. But you see here there are some impurities from ER, mitochondria, and early endosomes. But it seems to be a little bit different when we use Appli mode, but just trying to say that it's maybe not perfect, but it's as good as it gets. And when we compare that to the Oriya lysosomes, because they also use Western blot for quality control, you can see that there are really no contamination, and for instance, ER and mitochondria can only be found in the production waste. In this project, we also work together with Oriya because we want to actually compare the different isolation methods, and we are currently looking into a proteomic approach to understand better the protein levels that occur in both isolation methods. Nevertheless, when I apply it on the synchro patch, actually I can see very similar catching for the ultracentrifugation. But when we move on to the next step to whole lysosome configuration, you can already see that it's a bit reduced. Yes. It's a bit reduced and less stable. And if we apply the same quality criteria, we end up in something like close to 100 lysosomes that we have activated with PIP2 here and blocked with ATP. And when you see here the average trace, you can clearly see the nice response of PIP2 and blocked by ATP. And Oriya actually has received Christian's cell line, and they were isolating TPC2-YFP in the Oriya isolation method, and we can actually see the same functional response. And they have used Appli mode for enlargement, showing that both techniques, both enlargers can be used and give the somehow same functional readout. And capacitance was also not different, and also the current densities were not different. And finally, success rate are 10 to 20% lower when we use the ultracentrifugation, but still it's a reasonable amount of data, I think. And that's my conclusion. I think we can now say that we have established a novel method for organelle drug discovery in the native system that's complementing the sulfur technology. The method is fully automated. It can measure around 200 lysosomes in less than 20 minutes. It should be suitable for academic research, also for drug discovery. We have two sources commercially available with Oriya and also the ultracentrifugation. And we have functionally characterized 3 targets and found that the DCPI-BTUG mix is a nice tool compound, and the recordings at 35 degrees suggest a temperature-dependent regulation for TMM175. But besides lysosomes, there are so many organelles that also want to be patched. And we have now the first samples. We're working together with Oriya on mitochondria, where we applied the mitoblast suspension onto the recording chip, and we can see calcium-dependent current increases here, and they can be blocked by ruthenium red, indicating that we have measured MCU because the mycoblasts were MCU overexpressing. This is the last slide. I want to thank all the people involved, Oriya, Alexandre, Bastien, Marine, and Hélène, also Abdul Rashid. I forgot you to write you here. The whole Nanion team, in particular Nicoletta, who did all the beautiful recordings, and of course also Christian and his team, David is here, and also Nidish, who did all the preps and fluorescence and Western blots. And thank you for your attention.
so now we're on to our shorter talks. The first speaker is, could they come up please? There you go.
That's my extra slides.
Okay.
Just go on.
Great. So our first short talk is by Puravi Panda. She's going to be telling us about lysosomal calcium inflammation.
Thank you. Good afternoon, everyone. I am Puravi Panda, and I'm a DPhil student at the Platt Lab here at the University of Oxford. Today I'll be sharing about my research project, which focuses on the requirement for lysosomal calcium in the generation of the inflammatory response. So we discovered this by studying a rare genetic disorder called Niemann-Pick disease type C, NPC in short. As we all know, NPC is a rare progressive and neurodegenerative lysosomal storage disorder that's caused by mutations in NPC1 or NPC2. In NPC, we have impaired filling of acidic compartments with calcium, and this results in the accumulation of various types of lipids, as well as neurodegeneration and neuroinflammation, which promotes disease progression. Surprisingly, we see a skewing of the pro-inflammatory profile in NPC, where there is reduced production of IL-1 beta, but we don't see impaired generation of other pro-inflammatory mediators like TNF-alpha. And there seems to be a specific reduction in levels of pro-IL-1 beta, suggesting that there is impaired priming. The generation of TNF-alpha and IL-1 beta heavily rely on the NF-kappa B signal transduction pathway, which requires a stimulus such as LPS binding to its receptor, and this triggers a phosphorylation cascade where IKK, so I-kappa B alpha kinase, is activated. IKK phosphorylates I-kappa B alpha, which is an inhibitor of the NF-kappa B transcription factor, which is a P50, P65 heterodimer. And when I-kappa B alpha binds to NF-kappa B, it retains it within the cytosol. When IKK is active, it phosphorylates I-kappa B alpha, which is then ubiquitinated and degraded by the proteasome. And this frees up NF-kappa B to then translocate to the nucleus and turn on gene expression of target genes, such as TNF-alpha, which is released from the cell, and pro-IL-1 beta. Pro-IL-1 beta is the inactive precursor of IL-1 beta, and it requires a second step, which is an activation step, such as ATP binding to its receptor, which initiates assembly of the NLRP3 inflammasome complex. Caspase 1 is a component of the inflammasome, and it cleaves pro-IL-1 beta into its biologically active and mature IL-1 beta form, which is then released from the cell. So both TNF-alpha and IL-1 beta heavily rely on this NF-kappa B signal transduction pathway. Which brings us to the hypothesis that the skewing of the pro-inflammatory profile we see in NPC1 deficient cells could be because of altered NF-kappa B signal transduction. And to study this, we looked at the IKK-dependent phosphorylation and degradation of I-kappa B alpha, which is a critical regulatory step in the NF-kappa B signal transduction pathway. So I treated REW264.7 macrophages, which is a murine macrophage cell line, with U18666A, U-drug. U-drug is a pharmacological inhibitor of NPC1, and it binds to and inhibits the protein, mimicking the disease in vitro. I then stimulated the cells with lipid A, which is a bioactive component of lipopolysaccharide, and I did a time course experiment. And what we saw was that in the wild-type cells, we first see phosphorylation at around the 15-minute time point, but phosphorylation in the U-drug-treated samples is not quite to the same extent. And we also saw that there was a loss of I-kappa-B alpha at around the 15 to 30-minute time point in the wild-type cells, but in the U-drug-treated cells, this loss wasn't as significant. And quantification of the plots revealed that there was significant phosphorylation of I-kappa-B alpha in the wild-type cells, but not so in the U-drug-treated samples. And correspondingly, there was loss of I-kappa-B alpha, but this was not significant in the U-drug-treated cells, indicating that there is impaired phosphorylation and subsequent degradation of I-kappa-B alpha in the U-drug-treated cells. With the Western blot data, we could only get population-wide. We're looking at it at a population-wide level. So I also used flow cytometry to look at both individual cell-specific data as well as population-wide data. And flow cytometry results confirmed that there is indeed impaired degradation of I-kappa-B alpha in the U-drug-treated cells. Next, we wanted to investigate which particular feature of NPC causes this altered NF-kappa-B signal transduction. And given that we know that there's reduced filling of lysosomes with calcium in NPC, we hypothesized that the loss of lysosomal calcium homeostasis and insufficient mobilization of calcium could be responsible for this altered NF-kappa-B signal transduction. In NPCs, as I mentioned, there's reduced filling of lysosomes with calcium, resulting in reduced release of lysosomal calcium. And in another model, so the TPC2 channels, which we've all heard about, in TPC deficient cells, we have normal filling of the lysosome with calcium, but reduced calcium release. So in this separate model of lysosomal calcium, loss of lysosomal calcium, we would also predict that there would be altered NF-kappa-B signal transduction. And that is what we saw in these TPC2 deficient cells, where there were reduced levels of pro-IL-1 beta, but unaltered levels of TNF-alpha. And then I did the same time course experiment using TPC2 deficient macrophages, and we saw that in the wild-type cells, there is loss of I-kappa-B alpha at around 30 minutes to 1 hour. But in the TPC2 deficient macrophages, at 30 minutes, we still have significant levels of I-kappa-B alpha. But there does seem to be a delayed reduction or delayed loss at around 1 hour. So this is still a work in progress. I need more repeats to quantify it, but it suggests that there might be impaired and delayed degradation of I-kappa-B alpha in the TPC2 deficient macrophages. This brings me to conclusions. So we see that there's impaired phosphorylation and degradation of I-kappa-B alpha, and this could be contributing to the reduction or the reduced levels of pro-IL-1 beta in NPC1 deficient macrophages. And we also saw similar findings in the TPC2 deficient macrophages, suggesting a role of lysosomal calcium in the generation of normal physiological inflammation, pro-inflammatory responses. And currently I'm looking at the nuclear translocation of NF-kappa-B, as well as studying different steps of the NF-kappa-B signal transduction pathway. And I also hope to look at other signal transduction pathways in the future. I'd like to thank my supervisors, Fran and Nick, as well as members of the Plat Lab. And thank you all for listening. Thank you.
Excellent job. Questions? Yep.
You also look at the inflammasome activation?
I haven't quite looked at inflammasome because we're looking more at the priming step. And the inflammasome activation that's the activation step. And because we found that pro-IL-1 beta levels are reduced in NPC1, it seems to be the priming, the first part that's affected. So we haven't quite looked at IL-1 beta specifically, like the activation part of it. But I think that is definitely something that I could do in the future.
So regarding your hypothesis about TPC2 maybe being dysregulated, maybe in your model you could try and rescue the effects by activating TPC2. So we've got these agonists now available,
Right.
so that might be something to try.
Okay, thank you. Yeah.
Any other questions? No? Excellent. Thank you very much.
Thank you.
Okay, so on to our second short talk. And that one is Samantha Conster.
Thank you.
She's going to tell us all about EP2 receptors, the ins and outs.
Hi, thank you very much. I'm Samantha. I'm a second-year PhD student at Queen's University Belfast. And this talk's going to be a bit different from what we've heard so far because our group looks at GPCRs. So EP2 is expressed throughout the body and it's been implicated in a range of different diseases from neurological to respiratory to cancers. But to date, there's actually only one approved drug that specifically targets the EP2 receptor, and that's omadanemag isopropyl for the treatment of glaucoma and ocular hypertension. So clearly there's a real need here to try and find new and better ways to target this receptor. So EP2 is a class A GPCR, and we know GPCRs are obviously really important for drug discovery. So it's activated by prostaglandin E2 in the body, and then it signals via recruitment of its G protein, which is the G stimulatory protein, or GS, and that leads to increased production of cyclic AMP. Now, in general, GPCRs can also recruit beta-arrestin, and historically that was thought to lead to desensitization. So your G protein's displaced and then the beta-arrestin would stop the signalling effectively, and the receptor can then be internalized into the cell and then be either recycled or degraded. But we now know that actually it's a lot more complicated than that. So actually GPCRs can signal from a range of different locations inside your cells as well as at the plasma membrane. So from your endosomes to mitochondria, even your nucleus. And it's now known that signalling from these intracellular compartments can have completely different downstream effects than the signalling at the cell surface. So that's obviously really important for drug discovery. So for example, a different GPCR, the NK1R receptor, there were antagonists that did really well in the preclinical work, but once they got to the clinical trials for pain relief, they didn't perform as well. And it was actually then later shown by Nigel Bennett's group that actually if you used a membrane-anchored drug conjugate that could better target the endosomal signalling, actually that had better effects at sustained pain relief. So clearly it's really important to understand what's actually happening where in the cell with these receptors. So if we think back to EP2 then, it's slightly different in that actually it doesn't recruit beta-arrestin as strongly as a lot of the other receptors. So actually instead of it being an agonist-induced internalization, EP2 is being constitutively internalized into the cell. But the actual specifics of how this trafficking occurs and what it means downstream is still unclear. So the question that we're now starting to ask is, how do different agonists activate these internalized populations of EP2 receptors? So the work I'm going to present today is using an EB-BRET assay, so Enhanced Bystander Bioluminescence Resonance Energy Transfer. And we had a really nice introduction to BRET earlier. So the idea that if you have a bioluminescent donor and a fluorescent acceptor, when they're in very close proximity, we can get that energy transfer. And I'll come back to touch on the enhanced bystander part in the next slide. But the overall idea of what we're doing is we transfect HEK cells with plasmids for the different components we want our cells to express. So in our case, it's the EP2 receptor and then the biosensors. And then we seed it into our 96-well plates. Then after 48 hours of incubation, we can read our assay. So we have to add our substrate for our bioluminescent donor, so that's deeply C. And then you can also add your agonist to activate your receptor, and then we can read BRET on the plate reader. So if we look at the specific setup then that we've used, I mentioned that EP2 couples to the GS protein. So in our case, we're using a mini GS. So mini G proteins are basically an engineered form of the alpha subunit of G proteins. So that will be recruited to our receptor when it's in its active conformation. We have that tagged with our RLIC2, so that's our bioluminescent donor, our vanilla luciferase. And then we have GFP, and that's our fluorescent acceptor. Now in this case, we have GFP targeted to the plasma membrane using the CAX motif. But what you'll see later is that we can change that CAX to try and target this GFP to different locations throughout the cell. So instead of measuring like two different proteins interacting, we're actually measuring the recruitment of the RLIC towards certain locations. So if we have inactive EP2 like this, our mini G isn't getting recruited, the RLIC and GFP are further apart, and we have a low BRET. So where the enhanced bystander part of this comes in is that both the RLIC and the GFP are from the same Organelle species. So that means that there's a really efficient energy transfer. So even whenever they're far apart like this, you can still get some low level, and as those two components get closer and closer together, you get a really optimal transfer of energy. So if we now activate our receptor, we now get recruitment of this mini G, and now our RLOC and our GFP are in really close proximity. So now we get this improved energy transfer from the RLOC to the GFP, increased emission of the green light from our GFP, and an increase in Brett, which we can measure on the plate reader. Now, because we were interested in potential drug targeting effects for this receptor, we've looked at 3 different agonists so far. So we have our prostaglandin E2, that's our endogenous ligand in the body. Then we have evatanapag, or EVA. So that is an EP2 specific agonist that reached a phase 2 clinical trial for bone healing. And then we have the omadanemag, or OMD, so that's that clinical drug that we mentioned. So I'm going to apologise first for the quality of the graphs. I don't know what's happened with that, but I think you can still see the trends. So the first thing we wanted to do was try and generate those response curves. So if we look at the plasma membrane first, you can see that nice clear response to the prostaglandin E2 in the red. And then we have a lower Emax for both our evatanapag and our OMD, but very similar EC50s or potencies between the 3 different drugs. Now, I had mentioned that we could substitute out that CAX and change the localization of our GFP. So this is what we've done here. So we can use 5e or RAB7 to look at early and late endosomes respectively, RAB4 and RAB11 to look at fast and slow recycling, and we can also look at our Golgi. And we've been able to generate those response curves at each of those compartments. So I know there's a lot going on here. I just want to point out the main trends is that your orange curves, which is our OMD, that clinical drug, you can see this clear rightward shift in all of our intracellular compartments. And then while at the plasma membrane, we had the strongest response from our prostaglandin E2. At these intracellular compartments, there seems to be less of a difference, and in fact, it's only the evatanapag in blue that has any significantly different Emax at those different intracellular compartments. So if we look at it like this, it sort of simplifies it a bit. You can see much more clearly now our Emax on the left, the clear change at the plasma membrane between the prostaglandin and the other 2 drugs, whereas intracellularly, the blue, the EVA, is a bit of a lower maximum response. And then on the right, our EC50, you can see really clearly there how we have the significantly reduced potency of OMD inside the cell compared to when it was at the plasma membrane. So then we wanted to go on and look at the kinetics of this. So to do this, we've used the minimum concentration of each drug that was required to produce a maximum response at each of the compartments. So if we've added enough of the drug to get in, what do the kinetics look like? So we've basically measured the Brett using the same assay, but this time over a period of 20 minutes. So we can see here we're getting really fast activation of EP2 at all of these compartments, but where we are seeing differences is our area under the curve. So the amount of activation we're seeing at the different compartments is different between our different drugs. And a similar idea here with our other 3 compartments. And if we look then at our T-half values, so the time taken to reach half of that maximal activation, you can see sort of clearly here that it's fairly similar across the board between the different drugs. There's only a couple of significant differences. But do remember I said we're having to use the concentration of drug here required to produce the maximum response. So what this is basically telling us is if we have enough drug that it can get into the cells, we're getting really fast activation at all these different places. So this is supporting that idea of the constitutive internalization of the EP2 receptor. So where for a GPCR, where it's maybe agonist-induced internalization, you would expect really fast recruitment of the plasma membrane, but then a more delayed response in it getting inside the cell. So you would have a slower response at, say, the endosomes as it gets trafficked around. Whereas here we're basically saying, you add enough drug, it can activate all those different populations of receptors really quickly. So what does it actually mean then? Well, both the EVA and the OMD are non-prostanoid agonists. So if we look at our structures here, you can see they're relatively similar, but the omadanemag, we have a few more nitrogens, it's a bit more polar compared to our evatanapag, which has that bulkier tert-butyl group at the top. It's more hydrophobic, might be able to shield that carboxylic acid group a bit more. So from our data, it sort of looked like the OMD was struggling to get inside the cells, but then if you added enough to get it in, it could activate the receptor populations that were there. And this is actually supported in the literature by the fact that OMD is administered as the prodrug form omadanemag isopropyl. So it basically has an isopropyl group added onto the carboxylic acid, and that significantly improves the cell permeability of that drug. So that shows that our assay is measuring what we wanted to measure, but that's something we obviously want to look at moving forward, is how does the omidetipag isopropyl perform in our assay. And we're still at fairly early stages of this work, so in terms of the future work, we also want to try and visualise this EP2 localisation following different drug treatments using confocal microscopy. Then we also want to investigate what's happening downstream. So this assay basically tells you that you have the receptor and it's in its active conformation. It's not telling us what it's actually doing there, and that's obviously the really important bit for trying to target this for drug discovery. And then obviously eventually we would want to try and move into more disease-relevant models as well. So at the minute we use HEK cells because it's really easy to express these biosensors, it's easy to measure what we're trying to look for, but further down the line you obviously want to try and look at the more endogenous populations in disease-relevant models. So to summarise then, everyone here knows that it's really important to try and look at targeting specific organelles rather than just cells as a whole. And this is really starting to come to the forefront within the GPCR field now as well, that this is actually a really interesting avenue. And I've shown here that we can use Brett-based assays to try and monitor this spatially restricted GPCR activity. And then I've also shown that different EP2 agonists can differentially activate these internalized pools of receptors. And now the next stage is trying to understand what they're actually doing in those locations, because that could be really key for trying to target these receptors. And then lastly, I just want to say a massive thank you to my supervisors and my lab groups for all their support with this work so far, and BBSRC for funding this work. And a massive thank you for the Society for allowing me to speak today and share this work. And I'm happy to take any questions. Thank you very much.
Excellent, thank you. Questions?
Okay. So we don't have a tool for that in the lab currently. It's basically we've started with what we had tools for. So as well as the ones I've shown here, we also looked at the nucleus. We weren't able to measure signaling at the nucleus for this. We've looked at ER as well, same for that. So whether that is that our tools are not good enough to pick up the activation at this stage, or whether it's not there, is not entirely clear at this stage. It's something that we need to expand and look into further. Thank you.
From a physiological point of view, is it only liquid soluble agonist in the world, just kind of that's one question. And what do you think might be the physiological benefits of having internal activation
So if I go for that second bit first. So for different GPCRs, it depends on the GPCR and how it's internalised. For some, it's been shown that the signalling at the endosome site is a lot more sustained compared to the membrane. So if you're getting second messengers produced that are like closer to the nucleus site, that can have a bigger effect on the gene transcription. For some receptors, actually there's another one we've looked at in our group that actually seems to be the reverse, where actually you have a bigger pool of receptors at the endosome, but actually it's the signalling from the membrane that has the bigger effect on the gene transcription. So it's very GPCR specific. It's still a relatively new field within the GPCR field, trying to look at for which receptors does this make more of a difference and where should we be targeting it. In terms of the first part, yes, obviously if you can get things that cross the membrane and go into the cell, that's what we need to try and target those intracellular populations. There are different techniques. So Nigel Bunnett's group that I mentioned, they've done a lot of work on that, on how they could use membrane-anchored forms that then I think they used like a cholesterol type of molecule that they'd added onto it that helped that molecule traffic into the cell, and then it could target those endosomal receptors better than at the membrane, which originally wasn't known, which is why the first antagonist didn't succeed in those clinical trials. Thank you.
Jonathan?
Sounds great. Have you had any thoughts on data on fingerprinting, cell surface and intracellular different genes?
It's not something that we've looked at yet. This is what we need to sort of go on to the next stage now, is what exactly is it doing? What does it actually mean downstream? Thank you.
I have a question of general ignorance. So, receptors get into cells, endocytosis, that makes sense to me. I know how that works. How do these receptors get, for example, to the nucleus or these other compartments? Do we know anything about the trafficking?
Well, this is what it's still a fairly new field. We're still getting stuff out. So in general, for the endosomes, which is what our groups look at mostly, as you say, it's being endocytosed in and then can be trafficked around those different compartments. There is some evidence that sometimes you can get direct trafficking or retrograde trafficking, and that's how it can get to some of those other compartments. It's still unclear, and it's still unclear for the different groups of GPCRs how exactly it can signal from all those different compartments. So it depends also like what G protein it's coupled to. Do we have the right substrates in the right place? So what that actually means is what we're still trying to like look into.
No? Okay, great.
Thank you very much.
Okay, on to our final short talk. Slight change of schedule. Do we have any support?
They should be grouped in there. They might just be in the wrong window. There it is.
It's maybe because of the movies? There.
Ah, excellent.
This one. 1, 2, 3. There we go.
Okay, great. So our final short talk is from
Neuza.
Neuza, yes. Tell us about CI.
Yeah. Thank you very much. It's a big pleasure to be here presenting my work, lysosomes and nuclear communication, highlighting CLN3 function in these nuclear dynamics. CLN3 is a transmembrane lysosomal protein that is frequently mutated in Batten disease. Balabi's group showed that is important for the traffic of lysosome hydrolase to the lysosomes, from the Golgi to the lysosomes, also inducing lysosome tubulation and formulation. Also, it was recently shown that it's a chloride channel. And in 2022, Mounter's group showed that the mutation in CLN3 was inducing GPD's accumulation inside the lysosomes, impacting the whole cell lipidome. We recently published that by a transcriptomic analysis of CLN3 knockout cells that IAP was hyperactivated. YAP is a transcripted factor that is usually associated with the cell proliferation and inhibition of cell apoptosis. But here we show that it was inducing a pro-apoptotic phenotype. We show in 2 different cell lines depleted to the CLN3, and also using, in collaboration with Balabio, using brain tissue from the animals carrying the mutation most prevalent in the patient. So, a long story short, we show that c-Abl was the kinase inducing this alteration in YAP in condition of CLN3 depletion. So, phosphorylate YAP in this specific residue, the tyrosine 357, and this makes the interaction with P73, and both were contributing to this pro-apoptotic signaling in CLN3 depleted cells, also inducing a cell cycle arrest. We found that the cellular event that was behind this c-Abl activation was the increase in DNA damage. Also, by inhibiting the c-Abl activity using imatinib, that is a drug that is FDA approved, we could reduce the apoptotic phenotype, reducing the expression of these genes associated with this pathological effect. But how this CLN3 is inducing the DNA, is impacting the DNA homeostasis? This is our follow-up project. So we start by immunostaining lamine to understand how are the morphology of these nucleus. It's working. So by 3D reconstruction, I think you can easily see that CLN3 knockout has this increase in invagination, also by TEM microscopy, and in agreement with this altered morphology, we also observe increase in lamine expression when we deplete the CLN3. So because of this CLN3 ruling lipidome shown by Mounter, we wonder maybe we have some lipid profile difference in the nucleus that could justify this different morphology. So we isolate the nucleus, we perform lipidomic analysis, and indeed we found decrease in lysophospholipids, decrease in phosphatidylcholine, and increase in phosphatidic acid and diacylglycerols. We load these our cells with liposomes containing a fluorescent lipid labeling the heads, and we could see that in control cells, that is here. Sorry, I will use this because it was my dream to use this. You can see some colocalization between the lipid and the lamine, that this was not observed in knockout cells. So how this works, how lysosomes, or what this means at functional level of the nucleus. So in collaboration with Nuno Santos from Lisbon, we perform atomic force in the isolated nucleus, and we found that CLN3 depletion has an increase in Young modulus, so meaning that they have a decrease in elasticity. This is correlate, maybe, we are still studying a little bit better, with this increase in phosphatidic acid and diacylglycerol levels that we found in our lipidome results, and also the decrease in lysophospholipids. So I'm not showing here, but we also saw that these reflect in increase in nuclear membrane permeability. But how or what are the cellular function of lysosomes in nuclear interaction? So it's for to transfer some tablets. So using the same system, the liposomes with fluorescent lipids, we load the cells and we did a live image of both cell lines. And it was interesting to see that here you see the lysosomes that are coming to the nucleus and touching and rolling, and there is some transference of lipids to the contact area of the nucleus here that you can see by lamine overexpression. And in knockout cells, they are more static. You don't see this localization or like say, the lipids localizing with the lamine. And this phenomenon is even exacerbated when we overexpress CLN3. Let's see, it's working. So you see the CLN3 yellow, it's recruiting to the lysosomes, and they are doing these rolling events in tubulated lysosomes around the nucleus. And when we load the cells with the fluorescent lipid, now with rhodamine, you can see that the CLN3 is around these red lipids, and you can see some transference into the nuclear envelope. But how CLN3 is promoting these rolling events and these tubulated events around the nucleus? So for this mobility of the lysosomes, when they go to the periphery, they recruit kinesins. When they come to the perinuclear regions, they are normally associated with dyneins and dynactins. Consider all the literature, mainly the Balabio work about the interactome with CLN3 and this tubulated phenotype. We thought kinesin 5 could be a good solution that is involving tubulation and also involved in this peripheral transport of the lysosomes. So we overexpress CLN3 and KIF5B, and I think you can see here that the lysosomes containing CLN3 also have the KIF5B, and by immunoprecipitation of CLN3 we also could immunoprecipitate KIF5, so maybe they are interactor partners. But how these impacts the communication with the nucleus? So we immunoprecipitate KIF5 in both cell line, wild type and knockout, and it was nice to see that we have increase in the co-immunoprecipitation with KIF5 of NUP358, a component of the nuclear pore, and also NSPRIN2. But we want to know a little bit much more about this interaction, and we try to understand what was the motive that was behind this interaction in the CLN3. So searching in the literature and using AlphaFold, we found out that arginine 250 can be a good candidate. So we silenced, and we were able to reduce these co-immunoprecipitation of CLN3 with the nuclear components and also with KIF5B. Now, we are trying to we have the mechanism, and we are trying to fine-tune some details, but we are using this developed plugin in our group that I think we hope that will standardize a little bit this quantification of lysosome position, lysosome tubulation, lysosome, all the lysosomal characteristics that you can easily use in the lab. So by using a simple image, you can have the position. If you are using some label to the pH, you can correlate with the pH, or some parameters values has tubulated or not. And now we are applying this to several projects, including this one. So we are modulating KIF5 activity, and we are trying to understand if this is reducing the tubulated lysosomes induced by CLN3, and also we are correlate this with DNA damage in our condition. So we have the tool, and we have the mechanisms that we want to target. Take-home message. CLN3 is inducing these rolling events of the lysosomes around the nucleus, and by interacting with KIF5B, it's inducing some proximity between the lysosomes and the nuclear components, including the nuclear pore. And we think that this can be a useful tool to manipulate, and we are already collaborating with Maria João Amorim from GIM Lisbon to understand if this interaction is important for delivery of cargo to the nucleus. Thank you very much. Mainly to my group, Nunes Raimundo from Coimbra, also all our collaborators, and for you to listen.
Question?
You knocked down CLN3 totally, but did you, any other experiments, have any insight into whether the chloride reduction was relevant to the nuclear struggle?
No, because like the chloride, it was like two months ago, and we didn't know before that it was a function associated with CLN3. Yes, if now we will try to understand if it can be like a channel to modulate the chloride at the nucleus.
Is there anything special about where this is docking? Because given the size of the neutron envelope and the size of the lysosome, it can't be a bulk change.
I hope that will be in the nuclear pores. So I'm here to do CLAM to see if it's in the nuclear pores, because it will be a smart way to deliver a system.
You mentioned nuclear landing. I know people that have been nuclear landing that have theory here. Is there any link with that and also
Calcio, I don't know. It's a project that, CLN3 is involved with chloride, but we are also interested in laminopathies. Yes, it's currently a PhD student project that she just submitted to understand this communication under laminopathy condition. So if it's also impacted or not, if you can see like some metabolite transfer from the lysosome to the nucleus impairment. Yeah, we are looking on that.
I have a question. Do you think there's a role for contact sites between lysosomes and the nucleus in this?
The problem is you don't have the tether pair, but we are trying to use CLN3, but you don't have like good antibodies for immunostaining, but we are trying to overcome that and trying to optimize the PLA to show the contact sites. Also, I'm here to perform CLAM to understand if there is like a real contact site. Yeah, we are working on that.
Right. There's a structure in the yeast called the nucleus vacuole junction, and that's involved in autophagy. So my other question was, have you looked at starvation and whether that has an impact on any of your trafficking methods?
We didn't look to starvation to avoid the autophagic process in the all these dynamics, because you know, like exactly. So to avoid these, but yes, in the yeast, back into the yeast, I was trying to understand if there is some orthogonals, but you don't have mammalian orthogonals to the pair that they already show that vacuole exactly. No.
Thank you very much.
Thank you so much, Sandeep, and thanks for the organizer to put together such an inspiring program. I enjoyed it a lot, and actually I feel like a kid in a candy shop as a structural biologist when the speaker were comparing their favorite lysosomal membrane proteins that need to be characterized, so there's a lot of scope for the future. So today I will talk about lysosomal ion channels, which are most progressed probably in terms of drug discovery. So I talk about what we can learn from the available cryo-EM structures, and also what's going on in the patent space and in the drug discovery space. So I'm glad that yesterday we had already a great talk by David Yaslan about TRPM-L2, so I don't have to talk about TRPM-L2, but I will talk about TMM175, TRPM-L1, which is there are a lot of structures solved by Aztecs recently, and they are at very high resolution because we have fourfold symmetry, which really helps to get high resolution for cryo-EM. And then TPC2 is the closest to my heart, and it seems like a running gag, but this was a project I've done with Christian Grimm and David Yaslan in Munich during COVID. So starting out with TMM175, the first structures that were coming out are with these pore blockers that are relatively unspecific because they also block potassium channels. And here you can also see this is the density from cryo, and you can even see precisely what the orientation is. So they did MD to show that there are different poses that basically these pore blockers can assume in the pore. But what could be learned is that the pore architecture is actually different than in KV channels, so it's a bit broader. And then there was a nice follow-up paper by the same group 2 years later where they made a bulkier variant, so they extended this non-selective for aminopyridine with 1 or 2 rings and solved additional structures, and with this they went from 5-fold selectivity over KV channels to 26-fold, and they reached a sweet spot for these basically 2-ring system that is binding, extending towards the cytoplasm, whereas you get a different binding mode when you have the 3-ring system, but here you lose potency, and that's why so far the best balance between selectivity and potency is for this 2-ring system. So when we go to agonists for TMM175, people have found that there are small molecules that are actually for other membrane proteins. So there's LLRC8 antagonists, and there's actually for the GPCRs, the Swiss leukotriene receptor type 2, and there's also a K2P activator. So all of these have weak potency to activate the TMM175. And this tool called DCY1020 was derived from these LLRC8 antagonists, and there have been nice cryo-EM structures solved. So there's this optimized DCPIB derivative that only have a difference here, and here I've shown the structure with this hexyl ring that has slightly higher potency, and what you can see here that binds at the dimer interface between the 2 subunits of TMM175, and
what's also very interesting is that there's a potential arachidonic acid binding site in the TM4, M5, so there's a second agonist binding site, TAC819 is actually another drug that is a GPR120 agonist that binds next to a cholesterol molecule between TM5 and TM4, and that is an agonist as well. And what's interesting is that they are synergistic, so in green and in blue you see as individual agonists, they have a weak potency, but if you combine them, they have a synergistic effect on activation of TMM175. And when you look at the patent space, I've looked what patents are out there. So this is the tool compound we have just seen, the cryos, that has a tetrazole here. And then these patents are remarkably similar. So this is the Genentech patent that came out just last month. With 160 nanomolar, and this has an oxadiazole here, and then you have this, no, actually both of these have oxadiazole, so they're remarkably similar in their pharmacophore. So there are no structures about with these compounds, so we don't really know where they bind, but it's quite high potency, and it's impressive that they converge on a very similar pharmacophore. So this is actually basically what they have in common. They're all amino acids and have a material linker, and the variability is basically in the N cap here, and you have probably higher potency for these two. I mean, there's actually, I didn't find an EC50 for the Caraway Merck compound, but the Denali compound is threefold higher potency compared to the Genentech compound. So, and that brings me to these two companies are in Cambridge. Caraway was founded in 2018. Lysoway was founded in 2022. And Caraway had a collaboration with AbbVie in 2022 and was then acquired in 2023 by Merck. Lysoway has also an unnamed, undisclosed modulator, small molecule agonist. Most of these indications are Parkinson's disease. Here you also have lysosomal storage disorders. Lysoway also have on their website that they have a cryo-EM structure with the binding mode, and I tried to figure out from this blob where it is, and it doesn't really look like it's in the same position as the tool compound, so but it's a quite exciting space, and this is in IND enabling, and there's also smaller companies in the Netherlands, Meyer Therapeutics, and Tenvi and Threebrooks are also smaller biotech companies that have ongoing TMM175 programs. Yeah, I should mention, but we heard already yesterday that there is Parkinson mutation, like loss of function mutation in about 30% of Parkinson risk patients that have mutations, so it's a genetically highly validated target. So moving on to the triple MEL one, I just this is the compounds in Campbell, which is actually for triple MEL. It's relatively little compared to other ion channels. So although I would say that this is most advanced in these for these three lysosomal channels, there's still not so many different entries, and basically most are derivatives for ML from this MLSA1 that we heard already yesterday. There are actually, this is the publication from Aztecs that came out last year in Structure, and they have solved high-resolution structures around 2 angstrom, and resolution really matters here. So one lesson we learned from this high-resolution structure is the absolute stereochemistry for MLSA1, which is just a placement of one methyl group. So there was actually a previous paper, and I have this on the next slide. In 2017, the first structure had the stereochemistry wrongly assigned because at the resolution there you couldn't really see where this methyl group is, and now only at 2.5 1 angstrom, you could see that this is the R enantiomer that is bound here. This one is actually, they used a racemic mixture, and that's why they didn't really know what they have bound. And yeah, at this resolution, you just can't see if a methyl group is poking this way or the other way. So that's why a high resolution structure is really valuable in this case. The next lesson is also why is MLSA5 a higher potency agonist over MLSA1. And this is actually when they solved the structure, they could see that you have these an intramolecular hydrogen bond between the sulfonamide nitrogen and the pyridine hydrogen over here, and this improves the rigidity and has the benefit that there's no entropy loss when you bind it, because if a small molecule is very flexible and then gets basically immobilized in a pocket, it's thermodynamically not very favorable. So what medicinal chemists usually do, they try to rigidify the compound that is already in solution and has this intramolecular hydrogen bond. They actually did NMR at Astex and demonstrated that even in solution you have already this intramolecular hydrogen bond. Structure-activity relationship. So the original MLSA1 was having this indole group and quinoline group as well, and then the other compounds that were actually solved by Astex are all these bisulfonamide systems, and they have different substituents, so that they solved a few more structures. And what you can see here is that you have actually this indole ring. It's directed in 2 different 180-degree difference. So that's why you still have to solve a structure, even if there's only small differences, because here this sulfonamide is actually very similar oriented, if you see this, but the indole ring is flipped, and that's why, based on the chemical structure, you wouldn't, and if you dock it in, you would not necessarily come up with that pose, and these high-resolution structures are still useful to get the precise binding mode. So SAR is basically for these MLSA derivatives with sulfonamide substituents. So if you have hydrophobic groups, that increases the potency. You can see we are in the sub-micromolar range already for MLSA1. If you introduce this morpholino group, the potency drops. And that's why, I mean, they came up with this is the highest affinity or highest potency compound, and this is so hydrophobic that they couldn't solve a cryo-M structure anymore because they run into solubility issues. So that's a good example how in a drug discovery environment you can use cryo-EM to drive SAR and optimize compounds. And what's interesting, and we heard it already yesterday from David, is I think for L2, there is also only the enantiomer can already have a difference in agonist and antagonist for different MLs. So in the Astex paper, they kept PIP2 basically present in all the structures. And what you can see that both agonists and antagonists are actually going to the same pocket, and it is really the shape that drives if it's an agonist or an antagonist. So we saw yesterday with David that if you don't have PIP2 present, it's in a pre-open state, you only get full opening and full activation with PIP2 present. But they had PIP2 present just because it stabilizes the protein, also because they want to see if in the activated state you have actually full agonism and also antagonism. So the difference here between an agonist and antagonist, you see you have both this sulfonamide group, but here you have this dimethyl amine, which basically causes this round shape, and you have a more extended phenyl methoxy groups that is driving the antagonistic effect here. And if you look at the binding pocket, which is essentially the same, it sits here between the S6, S5 space at between 2 different subunits, so the light orange and the dark orange one, and can either drive activation or inactivation. You can see in blue you have the the pore closed when you have an antagonist which is elongated and pokes further into the space with the F513 and Y507. But then if you have this extra group over here, it broadens the pocket, and this is responsible for opening the pore via the S6 linker that is here, the lower gate, and causes opening. So there's also, interesting, there's also estradiol-derived antagonists that sit again in the same pocket and have this elongated shape. And yeah, it's actually quite unspecific what can be antagonists. There's also a TRPML1 mouse structure with sphingomyelin bound, which is a weak antagonist. So I think it's relatively unspecific what kind of pharmacophore you can have to make an antagonist here. And again, we looked into the drug discovery space. There's Calporta Therapeutics, another Cambridge company that was acquired by Merck in 2019. So they had a Niemann-Pick lysosomal storage disease program which was discontinued, and I mean we saw that they actually pivoted to TMM175. There's Casma Therapeutics, another Cambridge company, so they have a nominated drug candidate in 2025, which they progress currently, and then Lysoway Therapeutics, which we already heard of for TMM175, have also a candidate selected, and both of them have funding from the Michael J. Fox Foundation. And the Lysoway, they just announced that they will have their first patient dosed, I think, in May. So lots of exciting drug discovery going on here. But now actually the most interesting ion channel is coming up, and I think we have seen this slide quite a couple of times. I don't have to say much about what TBC2 does. So the reason, or basically the most important reason why I worked on it is because it has a role for virus release from endosomes during infection with Ebola and SARS-CoV-19. So there's a couple, during the lockdown, there were a couple of papers coming out. Also, there's one commentary here from Christian Grimm. Could an endolysosomal ion channel be the Achilles heel of SARS-CoV-2? And during lockdown, the university only allowed you to work on COVID programs. So that was golden times because I prioritized this project, and we had these these tetrandurin analogs from that Franz Bracher published a few years back. So that there was a talented PhD student called Susanne Gernt, and she made these synthetic analogs of tetrandurin that are more soluble, so SG094 and SG005, and this is in-depth characterization of the SNR analogs that they did, both in P mode and in N mode, so both enantiomers are actually active, and these inhibitors basically suppress TBC2 activated by both synthetic agonists, and it's also both enantiomers. So we tried to get cryo-M structure, but only succeeded with the S and R. SG94, for some reason we couldn't get, tetrandurin is not very soluble, and SG05, we didn't succeed to get a cryo-M structure, but we had both enantiomers bound to the same site, which is here in the voltage sensor domain. And it was very interesting because it's an asymmetric binding mode. So this is a homodimer with two identical subunits, but we only saw one antagonist molecule bound in the voltage sensor domain. And the structure of it becomes asymmetric. So here in this crevice between the S6 and the voltage sensor domain, the space enlarges by the presence of this antagonist, whereas on the other side, the empty pocket is because the space gets smaller and there's no space to bind a second molecule. So we have also mutated the two main aromatic side groups, F55 and Y436, and then we lose binding or inhibition when we mutate this. So this is a Fura2 assay, and this is actually the purified protein. You can do melting curves, and we lost the stabilizing effect when we have this mutation, confirming that this is the right binding pose. And what's interesting is, if you are familiar with voltage sensing ion channels, the voltage sensor domain, which is actually silent in TBC2 because it doesn't have 4 arginines that are sensing the membrane potential. But what this molecule does, it causes a downward shift in the S4 sensor, just like in a voltage-gated ion channel, and moves it into a downward position that basically prevents pore opening. And yeah, I mean, there was actually, when it got published, it was a nice surprise. Someone wrote a commentary on it. I think must be the reviewer who also was fascinated that we have here what I call the Achilles heel of TBC2, that you have a pseudo voltage sensor domain that's inactive, but that actually is the drug binding site that blocks the ion channel. So yeah, we had a commentary on this, and it's quite an interesting binding mode. That we didn't expect to see, but it's fascinating, and it makes it quite different from the other lysosomal ion channels which don't have voltage sensing domains. So what's also interesting, we did these melting curves which are a bit messy, but the red curve is actually, when you incubate it with PIP2, you get a thermostabilization, and when you incubate it with PIP2 plus the antagonist, you prevent this stabilization. So we think there's an allosteric effect that blocks the PIP2-induced conformational changes, and this is just an overview on the tetramer, where you can see that the binding of the small molecule in the voltage sensor domain causes a change in this S4, S5 linker, and here these are superpositions with the open and closed structures that were published before, showing that you stabilize it in a closed state in the presence of the antagonist. And that brings me to the end, and I hope that you have seen that there's a wealth of recently published cryo-EM structures on the 3 different ion channel families, and they shed light on the druggable pockets and illuminate the mechanism of action for small molecule modulators. And there's a lot of biotech companies and probably also big pharma, and they use that for the drug discovery pipelines, structure-based drug design to advance new drug candidates into towards the clinic. And thank you for your attention. I'm happy to take that.
Lovely talk. I was wondering, it's similar to the A and P modes or the N and P modes in TBC2. Do any of these modulators of T9175 have any preference for a cation versus proton flux?
I actually don't. I mean, I'm not sure. These papers were mostly on cryo structure. There wasn't so much functional data, but I don't think that I'm not aware that there's a big effect like this. I mean, TRPM-L1, so with TMM175, let me think about this. They haven't, I think there's a controversy in the field, which is complicated. And
that's where I'm coming
yeah, exactly. But I don't think that, I mean, most...
So NASDAQ gave a talk about
yeah.
San Francisco, and they seem to prefer measuring proton flux to TMM.
Yeah.
Other people do measure the transients.
And so, maybe their patent was then basically benchmarked on proton conductance. No, I mean, this is something, it wasn't really covered, and I just started working with TMM175 myself, and I think there's a few mysteries there that needs to be solved. Yeah.
So, for biological reasons, how hard do you want to hit your targets for any curative disease? Because everyone is talking about, okay, we want to, it's a regulated system, the lysosomes. So I can imagine that you don't want to hit them full force.
Yeah, I think that's actually a great point, and I think we had actually good discussions in the pub also. Some people think, TMM175, there's gain of function, loss of function mutation for Parkinson's, is that actually a good drug target? But I have to say that, Martin Gunter was here, but I worked on other ion channels that have to there can also be gain of function or loss of function if the threshold for activation shifts, but then you can maybe fine-tune it just where you want it to be. So I think that's the beauty about ion channels, that it's not necessarily switching on and off, but you actually fine-tune it. And also if you have allosteric modulation, so a TMM175 has even these 2 different binding pockets that work synergistically. So but I think that remains to be established how much you want to activate it. And I mean, for the TMM175, there was interestingly also a paper that showed that basically in vivo they are more potent than they seem by in vitro characterization, and they argue that they are accumulated in the lysosome, which is maybe one strategy people would use for drug discovery, that they make them basic, that they get basically trapped in the lysosome, they have a high local concentration, and then you're only on target where you want to be. So I mean, I'm more structural biologist, so I'm not the best person to ask this question, but I appreciate that you think I'm the expert for this. Yeah.
Thank you very much. Just out of curiosity, depending on these channels, have allosteric binding sites where endogenous ions bind, sort of sodium. For example, this is a thing in GPCRs, but I'm not really aware that for this, but I think there are allosteric sites. So there also, I mean, we had already Rebecca talking about TPC2 and the agonists, and actually there's a lot of lipids. They're not only PIP2 in the density maps, we see several lipids. And I think there's actually, for these ones, and you saw there was for one of them a cholesterol bound under the agonist. So I think for the ion channels, it's less about allosteric ions, but more about allosteric lipids. And this was also like a potential arachidonic acid binding site in the TMM175. So I think this is one area I'm particularly interested in, because I think that is really important for, you have to see the lipids almost like a subunit of the ion channel, and they are often overlooked, but now you see them in the structures, you don't know what they are, but you have to do lipidomics to understand precisely what they are, and if they are maybe also activate, or basically the lipid ratio changes through metabolic state changes. I mean, many of these are arachidonic acid is also a signaling lipid, right? So I think that's an area you have to look into, and also you might be surprised if your drug candidate doesn't work at the end, because maybe you make it in a test tube and you overlook what is the lipid environment when you enter the disease condition, which, So I think that's one important area that I think will be very important to make efficient drugs for these lysosomal ion channels. And also people have to be careful about retargeting to the plasma membrane. And we will continue to appreciate the lysosomal patch technology, right? Because the lipid environment is very different in lysosome than the plasma membrane. Thank you.
Mm-hmm.
Nice work, nice summary of all the structures, all these targets that you have shown. So there is a general high potency on the TMM175 with the TMM1 analyzed by such that this is a limitation on the TMM1 to respond to what is with the TMM175, or do you see any additional interaction in this molecule on the binding side of the TMM1 to increase tenfold what is the difference between TMM175 and TMM1?
Sorry, was it about water? So what? I just was. I think there was the door, and I didn't really fully capture the question. It talked about water molecules.
When you look at the binding side of the TMM1 with respect to the TMM175, do you see any possibility to make an additional interaction of this molecule with the TMM1 to try to increase the potency, because there is a tenfold difference in the potency?
I mean, I haven't really spent that much time. I'm not a medicinal chemist, but I mean, I think the TRPM are already quite potent, and the TMM175 weren't super high potency. But I think it's interesting because you have this synergistic effect, right? And then again, TMM175 is so complicated, because then how do you define your EC50 with the proton conductance or potassium conductance, right? So I think that's another complication. But I haven't looked in detail at that binding pocket and if there's a possibility to improve it. But I also think that maybe these patterns are not binding in this DCY. It looks they might bind somewhere else.
Okay.
Yeah.
let's leave it there.
That's a Martin.
One very quick question.
Yeah, okay. I'm trying to ask the same question I asked Jonathan yesterday, whether you looked at the binding proteins and JP2 or CLSM12 in conjunction with this, the structural work you've done here.
No, not yet. And I was basically, I have. This is actually a few years back when we did COVID-19. So, but I think that's a very interesting area, right? Because no, we haven't done it, and I think we first would have to characterize maybe the interaction of NADP with these unstructured proteins alone, which I find also a bit difficult to work with. But then, interaction. I haven't tried to make an AlphaFold prediction, but if you have such an unstructured protein, it's very hard to think where that would interact with the.
Have you even tried the DSF approach there, or would you contemplate that?
You can try it, because sometimes with AlphaFold you get interesting effects if you combine with the AlphaFold multi or the Bolt server, where you can have multiple proteins co-folded, and sometimes they get structured when they have the right interaction partner. So actually I haven't played around with that. It's worth giving it a try, but I think the challenge I see is also, and I mean, the speaker from yesterday, I said actually there could be PTMs in these unstructured proteins. You have a lot of phosphorylation sites, so in the AlphaFold I currently cannot mock up phosphorylation, so I will not have the realistic interaction, because that's a one of them was very basic, but as soon as you have phosphorylation site, it's not going to be that basic anymore, and to predict the interaction with TPC2 correctly, I have to know what is the physiologically relevant PTM and mock it up, basically. So, but this is a lot of the mechanistic questions we still have to answer and look at the PTMs. Maybe you can mock them up with phosphomimicking, phospho-knockout mutation, see if that makes a difference in the interaction, and then slowly understand how this works. But I think it will not be as simple as, getting these unstructured proteins and at TPC2. Otherwise, someone would have already solved and had a nice high-impact paper with the full complex.
So the quick answer to that one. Right, I think we'll leave it there. Thank you.
Yeah.
Thanks again to both of our speakers after the break, and all speakers this morning as well. It's been excellent. Another round.
Well, thank you all for coming. I think it's been a really fantastic day and a half. We started off yesterday hearing about clinical trials from Fran Platt, and we ended up with Katerina this morning. We're talking about the detailed molecular structures and interactions with drugs. And over the day and a half, we covered every level in between. So I think it's been a fantastic day and a half. As I indicated, I think this is a unique, new type of meeting. The most important thing fostered by the BPS is to bring together a community of people interested in organelle pharmacology, and I think we've been hugely successful in doing that. I don't think there's anyone here that can doubt the importance of targeting organelles. It's a way of targeting diseases with perhaps more precision than that we have the capacity to do by perhaps modulating these proteins through cell surface receptor signaling pathways. But then we've heard today that some of these receptors actually are residing in these organelles, and this is perhaps another new frontier for organelle pharmacology. And as Jonathan Marchant reminded us, perhaps we mustn't forget the membraneless organelles, and that's perhaps going to be a new area in how we can modulate those with drugs. I think one of the reasons this field has been advancing so rapidly is the development of new technologies, not only the advances in structural biology, cryo-EM, but also the fluorescent technologies, fluorescent proteins for looking at functional biology and their modulation coupled to screening protocols. But, we've heard today, and largely due to the work of our sponsors today. We've heard from Oriya, who are developing pure organelles off the shelf, which will be useful for drug discovery in this area. And we've heard from BSIS, Sofian, Nanion, who have developed techniques for looking at these ionic events deep in the organelles. And I never thought that would be scalable for screening, but it now seems that's possible. So I'd like to thank you all again for coming. I hope this is the first of many meetings in this field, but it just leaves me to have perhaps the most important thing, and that's to recognise the importance of the early career researchers who have produced posters and presented excellent oral communications. And our esteemed Vice President Meetings, Professor Mark Evans, judged the oral communications and posters, and we have some winners. And I will announce them now. So for the best oral presentation of this meeting, the prize goes to Samantha Collister of the Queen's University at Belfast. Congratulations. I think you've already been emailed your certificate, and I'm sure that the money will follow. And the runner-up for the oral presentation is Nusa Dominguez from... Are you here? Are you from Coimbra? Yes. Yeah, excellent. The best thing about Coimbra is there's a road highway called IP3 that runs. Oh, you're stick. There we are. Your dreams come true twice today. And finally, for the best poster, it goes to William Burton of this department. Congratulations. Come on. Okay. Well, I think that's the end of the formal meeting. Thank you again for coming. Let me just remind you once again, for those of you who are here tomorrow, we have an Oxon seminar here at 11 o'clock from Sabina Teherovic on endolysomal dysfunction, road to neurodegeneration. And I think it will be recorded, so if you have to fly back somewhere, you can always log in after tomorrow. So thank you again for coming, and hope to see you again soon at another meeting or back here in Oxford. Thank you.
Yeah, absolutely. I learned a lot about that.
Oh, yes. I'll take the hand. Lovely to see you. It's a very nice meeting. Yeah. Yeah, take that. Bye.